Everybody, it's my pleasure to introduce Trang for her oral defense. I think we have all the committee members and many audience members here for the general session, which I think is going to last between 30 and 40 minutes. And there'll be a time for questions at the end, but we'll just let Trang give her oral presentation. Before she starts, I just wanted to personally introduce my work with Trang has been quite amazing. She's an amazing scholar who came with cum laude from Vietnam, who came to Harvard to learn about neuroscience and psychology in some of the most amazing labs there, and was recruited into Stanford Graduate School of Education for a PhD right around the time that Stanford was starting to make major commitments for the connection between neurosciences and education. And Trang's dissertation wound up focusing on two very powerful ways of combining these. One was forming a collaboration with the Norsha Lab around using powerful new oscillatory paradigms to look at brainwaves and study brainwaves to see how kids' brains are changing over the course of education, and at the same time, forging new research practice partnerships with very innovative schools, schools that span from elementary to middle school, to embed a state-of-the-art neuroimaging lab inside of a school and make it part of the school, and collaborate with the school about what kinds of questions could we answer together as educators and as neuroscience researchers that would be both rigorous in terms of their methodology and theoretical impact, as well as meaningful to the school. And as we built up this research practice partnership, Trang was at the very beginning, at the very early stages of us forming the prototypes, forming the questions. And the questions we wound up really focusing on is what's changing so powerfully in the minds of the kids as they progress from the earliest stages of the school all the way up through when they complete in 8th grade. We know that there's profound changes that occur. We know that we can measure these changes, perhaps the best at the level of how the brain internalizes the symbols of language, the written word. And so Trang is going to be presenting some of the very first data that was collected at this school systematically to answer scientific questions, to push the edge of what we could do with these powerful oscillatory paradigms and what we could do in the context of a living, breathing school where the participants were participating inside of their own school. So without further ado, let me introduce Trang, and thank you for many amazing years collaborating together. You're one of the most persistent and devoted students I've ever worked with, and it's been a pleasure. So thank you very much. You've honored our lab, you've honored the entire kind of collaboration with your hard work over many years. And we're looking forward to seeing some of the scientific results that you believe in addition to all the other amazing discoveries we've had.
Okay, thank you, Bruce. And thank you everyone else who joined. I'm so excited. So this is my dissertation defense talk. I'm going to be talking about how individual variation in the brains of developing children. I will start with giving you some exercise, some work for you to do. On the screen, I'm going to be showing you a series of letters and symbols at the rate of 3 per second. And then after all of that is being shown, it's going to go blank. I want you to count the number of times you see something get bigger in size. Don't overthink it. I know that there are some like overachievers in this group here. These changes should be pretty obvious, okay? And once the set is done, please type in the chat the number of times you saw a size change. The first person to get it right will get a badge of honor of love and respect from me. You ready? Let's get started. Okay, go. Answer in the chat. Let's see who is first. Oh, Tanya, yay! Okay, I love you the most out of everyone in this group. So, while I had instructed you to notice size change, you possibly were going above and beyond, and perhaps even doing some reading of these words. This is pretty normal in adults. Reading is a very automatic process. Literate adults, what? Literate adults can read rapidly and effortlessly at a rate of approximately 5 words per second. Reading can be so automatic that it's difficult to refrain from. This leads you to just automatically read the words on the screen, even though you might not be intending to. Just the presentation of the letters or words alone can activate a whole network of language-related areas. Looking at this video, your brain is firing like crazy, rapidly recognizing that the words are being presented in the midst of what looks like gibberish symbols. Even if you didn't want to, your brain couldn't help but activate this reading network. But to be able to reach this level of automaticity that we see in adults, children need to learn. Learning anything, learning to dance, learning to read, learning to recognize faces, requires the combination of two important but distinct processes: learning the elements and learning how to configure the elements into new forms. You can think of the difference in elemental learning and configural learning as the difference between learning the features of the face, like the eyes, the nose, the mouth, and how learning how these features combine together to form familiar faces that you recognize, like in the audience today. Hi, Blair. When a child learns to read, they rewire the brain to respond to the elements of reading. In our English language, it's the 26 letters in our alphabet. Letter form processing is the brain's ability to discriminate between letter-like letters and non-letters. This seems to be the easy part. Nearly all children learn to recognize letters, but the hard part is if they're able to configure the letters together to create whole word forms that carry meaning. This is referred to as word form processing. It is a recognition that this visual stimuli is a word, while this is a non-word. It is this configural learning that seems to be harder for children. Many children struggle to learn to automatically recognize the thousands of visual words in our language. Since these two learning processes are so integral in helping children reach the level of reading expertise and fluency found in adults, it is important to identify how they evolve as they learn to read and to figure out early on which children might struggle with these processes. So how can we study how these processes develop? How can we identify early on which children are struggling? How do we do it way before the children are even able to verbalize and tell us that they're struggling? Well, by looking at their brain, of course. What do we already know about the children's brains and how it changes as they learn to read? We know that in order to read, they need to first recognize that they're seeing something on the page. Any visual stimuli that is coming in is immediately sent to the back of the brain in this area in the back that processes visual information. Then, children will need to recognize that some of this visual stimuli are letters from our alphabet, or even configure these letters together to form words. The visual word form area, or the VWFA, shown here, developed to be increasingly sensitive to letters and words over other forms of stimuli, like faces and places. This development occurs through increased exposure to letters and through learning. The VWSA is the area that is predominantly implicated to be involved in letter form and word form learning that I described earlier. But learning to read does not stop here. After recognizing word forms, children need to learn that different words hold different meaning. As they accrue more familiar words, this green network that you see here also matures and changes. Reading is also a phonological process. When you're reading something visually on a page, you're also hearing what the words sound like in your mind. Children's brains have to grow attuned to this integration between the information that they see on a page with what the words might sound like. What's more, on top of this, where the children places their attention also affects how they process the reading information and thereby the development of these regions. All in all, the reading brain is a complex yet integrated network. It is a complicated neurological dance as different parts of the brain are trying to work together. This gives rise to a lot of opportunities for individual variation at different nodes of this reading network. Thus, my thesis calls for a better understanding of how the reading circuitry is changing over the years as children learn to read. Individual variation is in different parts of the circuitry could have huge consequences in the children's lives, since reading is such a critical skill for their academic and career success. Thus, for my talk today, I will share with you two of the studies that I've done that focuses on the early processes of learning to read, specifically the processing of early visual information, letter forms, and word forms that are typically found to be related to these two nodes in the reading network. I will be showing you the different dimensions at which the brains of children in 3rd to 8th grade vary in the way they process information. In the first study, I will show you how individual variation in the amplitude of the neural response to a word reading experiment is related to the age of the children. So I will be showing you how younger children might have different amplitude responses than older children. Then in the second study, I will extend on this work and show that individual variation in the latency or the timing of this neural response is related to the children's reading abilities. I'm going to show you how the response time of poor readers might be different from better readers. Together, these two studies allow us to simultaneously measure different dimensions of individual variation in children's reading brains, thereby giving us powerful tools that would be critical for the future development of early assessment or even diagnosis of reading disorders. So let's start with study 1. The main research questions that I asked included: 1, can we detect letter form processing in 3rd to 8th grade children? 2, can we detect word form processing? Question 3, are these how do these two processes function separately versus together? And question 4, are these amplitude responses correlated with age? To investigate how children's brains process letter forms and word forms, researchers often use a method called encephalography or EEG. EEG measures the brain's electrical signals using 128 sensors placed on the scalp. Participants wear this net of 128 sensors while looking at something on the screen or while doing some task. The raw data that comes in looks something like this. Each of the lines you see here represents the data received at one of the 128 sensors. Data is being captured at a rate of 1,000 times per second. A very powerful EEG technique called the steady state evoked potential paradigm, or SSVEP, has been used to study letter form and word form processing. I've already shown you snippets of it, but here is exactly what it is. In SSVEP, visual stimuli is presented at a specific frequency. In this study here, it's presented at a rate of 10 items per second, or 10 Hertz frequency. As the brain is seeing this visual stimuli, the brain is entraining to that frequency. And so if visual stimuli is presented at 10 Hertz, the brain is also pulsating at the same rate of 10 Hertz. This 10 Hertz frequency of visual stimulation is what we call the base frequency. But you also see here that you have words interspersed within a sea of pseudophons. This occurs 2 times every second, and it is what we call the deviant frequency, because it is the frequency at which a deviant stimuli is being presented. What you're seeing here is actually the first study that utilizes SSVEP to study the brain's automatic letter form and word form processing in adults. They intersperse words within pseudophons and words within non-words. SSVEP is a particularly suitable approach to measure automatic discrimination of categorical change, as it allows us to study signal responses to visual stimuli presented at a specific frequency. When analyzing the SSVEP data, instead of using the messy waves that you see in the previous slide that is full of noise, SSVP allows you to just pull out the frequencies of interest for analysis. So in this study, Loki et al. Was able to show significant letter form and word form processing in healthy adults. SSVP has some advantages, but also some limitations. So an advantage it has is that it can produce very robust signal response with high signal to noise ratio. This is because we're analyzing only the specific frequencies that is related to the stimuli that is being presented. We're getting rid of the outside noise that you find in other EEG studies. This allows us to shorten the data collection to just a couple of minutes. If you've ever worked with children before, you know that they have a really short attention span. So anything that we can do to shorten the study period is ideal. It does not require explicit stimulus processing. What this means is we're not asking the children to read. We don't have to. This is very helpful because then we can work with even younger children or even children who are not able to read. SSVP studies have also shown that letter form processing signals are consistently found across studies that are studying adults and children. However, inconsistent evidence have been found for word form effects in adults and in children. So in adults, this is a paper that I also worked on that we showed that we were not able to fully replicate Loki. And in children, no SSVP studies today, or when we were working on this project, found word form effects in children. So this leads us to our study. We know that SSVP is very sensitive to letter form processing, but relatively insensitive to word form information. Thus, SSVP should be developed further. And we have reason to believe that the 10 Hz frequency that Loki et al. Used may be too fast for word form processing. Let's try this yourself. I'll show you a series of words and non-words on the screen, and please tell me how many words you see. So after this is done, just type in the chat how many words you can actually count. Okay, let's see. Go for it. It's hard, right? Wow. None of you got it right. You're all wrong. Fail. No, so it's really hard. If it's hard for you as adults, it's going to be very hard for children. So then we decided that we needed to change the presentation speed if we were to study children. What we know, we made the decision of what presentation speed we're going to use based on what we know in previous literature. We know that word form processing responses in children typically emerge between 200 and 500 milliseconds. This tells us that our rate of presentation or our base frequency should fall between that range. We also know that Dr. Yateman and Dr. Norsha, who are here in the audience today, published a paper that showed that text selective responses were maximal at 1 Hz when they did an exploration across multiple frequencies. This leads us to believe that we should probably use a 1 Hz deviant frequency. And then in a paper that our group published, we found that when we slowed the presentation rates down, it evoked stronger neural response in the study of adults. And so we decided to use a 3 Hz base frequency and a 1 Hz deviant frequency. We also changed the task. So Loki et al. Used a passive viewing task of a cross. They asked participants to just look at a cross in the middle of the screen. We decided to change the task to a size change size change test. Basically the one that you did at the very beginning of the presentation where you indicated the number of times that the whole visual stimuli changed in size. We decided to do that because then it would put the participants' attention on the entire word form instead of just a little cross in the middle of the screen. So this is our SSVP paradigm. We use a deviant frequency of 1 Hertz and base frequency of 3 Hertz. For condition 1, we put words in a sea of pseudofonts. So it appears as pseudofont word. And then condition 2, we have non-words in a sea of pseudofonts. Condition 3 is words in a sea of non-words. This allows us to study the contrast between when pseudofonts appear versus when a word appears. The difference between this can give us the letter form processing signal. Whereas for condition 3, the difference between a non-word and a word gives us a word form signal. Like the child's brain is able to detect that, oh, I'm seeing a word now, it has some meaning. And then condition 1 has both letter form and word form processing because the child's brain is able to detect, oh, we're seeing different like letter and non-letter here. At the same time, the letters like configured together and form a real word. So both of them is happening at the same time. This allows us to, this setup of 3 conditions allow us to study not only how letter form and word form processing occur separately, we also see how they function when they're happening together. Okay, so let's get straight into the results. For question 1, we asked if we can detect letter form processing. We know that the VWFA is typically involved in the processing of letter form. I've already shown you that it's found in adults consistently, it's found in children consistently. So we expect, yes, of course, we would also find a robust signal at the individual level in children. We did this by analyzing the response at the deviant frequency for condition 1, which is the word versus pseudofont contrast, and condition 2, which is the non-word versus pseudofont contrast. And this is what we got. Using a method called reliable components analysis, where we're able to pull out brain regions that are reliably getting the same neural responses, and they have similar responses over certain timeframes, we get this brain region here. With the source of the signal being highest around this yellow region, less in the red region, and pretty much non-existent in the blue region. To orient yourself, this is a top-down image of your brain, with this being the nose up here, tiny little nose, and the back of the brain. This region in yellow, if you're able to turn this brain, you will see that it is very similar to the VWFA area. And then when we look at the amplitude of the signal response that is coming from this brain region, we see that both condition 1 and condition 2 are significant. And by significant, I mean that the amplitudes are significantly above 0. That means that the brain of these children are able to detect differences between letters and pseudofonts. The amplitude of the condition 1 and condition 2 are also significantly different from each other, with word versus pseudofont contrast being more like higher amplitude than the non-word versus pseudofont contrast. And we believe that this may be due to the fact that the word versus non-word contrast also has some word form processing, like I mentioned before, and we will explore this later. For my second research question, I asked if we can detect word form processing. Word form processing also occurs in the same VWFA area. I've shown you that the signal is inconsistent in adults, and I've talked about how the signal was not found in children in the past. And so what we expect, based on the changes that we made to the paradigm, we believe that we can find word form processing in children. We did this by analyzing the response at the deviant frequency for condition 3, or the word versus non-word contrast. We get the same brain region as the one found in the previous slide, and we find that the signal that we got is significant. Yay, we got the word form processing signal in children. When we compare this with the amplitude of condition 1 and 2, we see a clear condition effect. So this is the highest, and then this one, and this one. This high significance that we're getting, the incredibly low p-value, each of this, this is basically showing that using pairwise t-tests, each of these conditions are significantly different from each other. We're getting high significance, incredibly low p-value of p of less than negative 0.001, very small error bars within each of these amplitudes. This result just seems way too good to be true. And so I had to check and make sure that I'm getting something real rather than just some coincidental result. And I did that by correlating the amplitude between all the different conditions together. What we found was this is comparing condition 1 with condition 2. This is condition 1 versus 3, and this is condition 2 versus 3. All 3 of these are significant at a p of like 10 to the negative 5, 10 to the negative 11. This is like incredibly low p-values. This is basically showing you that children who have low amplitude results for one condition also has low amplitude results for another condition. This confirms that our findings are not coincidental, but that the children's brains are truly responding to the visual stimuli that is presented, and that the word form processing signal that we find for the first time ever in 3rd to 8th grade children is real. For the third question, since I did get a word form processing signal, I asked whether the letter form and word form processing, like how they function when they are separate versus when they're together. So as I had mentioned to you before, condition 1 might have both letter form and word form processing. Condition 2 only has letter form, and condition 3 has only word form. When you do a little bit of simple math, you're going to realize that when you take condition 1 minus condition 2, you're going to get condition 3. You take letter form processing and word form processing, you minus the letter form processing, you'll get the word form processing. So we wonder whether, this is the same graph from before. We wonder whether if you take the red bar, you minus the green bar, if that is going to be equal to the blue bar. This blue bar is the same blue bar, it's just scaled differently. And then so I took the difference and I find that they're not actually the same. I find that actually the word form processing that is present in the word versus non-word contrast is significantly higher than the word form processing that occurs in the word versus pseudofont contrast. So this makes me believe that the context in which words are presented is important. When words are embedded within a sea of non-words, perhaps the brain is more attuned to the detection of word forms, because it's seeing, oh, it looks something like a word. They need to the brain needs to be more activated to process words. But when a word is presented within a sea of pseudofonts, that activation doesn't need to be as elevated. And in fact, the key findings that we see thus far, this is the same graph from before. This finding, we continue to explore a little bit more, and our RCA approach allowed us to pull another brain kind of filter brain area that also had similar results. For the fourth question, we wondered whether these amplitude responses correlated with age. Since we got two distinct and reliable sources, we wondered whether the brain signals from these two sources differed in their relationship with the child's age. When we run a correlation analysis between the child's age and the amplitude coming from that brain region, we find that the second brain region is correlated with age. The P is very small here, but it's not correlated with age for the first brain region. This basically tells us that for younger kids, they have a higher activation than older kids. When we look at condition 2, we see the same thing. Second brain region has a correlation with age, and the first brain region doesn't have a correlation with age. So what is your prediction for the third condition? The same, perhaps. Well, actually, it's not, and it's quite the opposite. We see a correlation with age for the first brain region and not for the second brain region. In all three significant findings that you see here, the ones that I have circled in black, they're all showing that as the child's age increases, their amplitude decreases. We believe that this might be the child learned to read, and so their processing of reading related information is less taxing over time because they're more experts. So the amplitude decreases with age. And then in terms of figuring out why there's a difference between brain region 1 and brain region 2, what we think is happening here for those neuroscientists in this room, we believe that the second brain region is very similar to the P300 signal that you see in EEG studies that is involved in the detection of coarse differences and between different stimuli. So just coarse differences that is general, like it's not specific to reading related, just coarse changes. So that's why there is a developmental change when you're looking at word versus pseudophone and non-word versus pseudophone contrast. But this brain region up here is more similar to the N170 brain region. And N170 is more specifically related to reading information. And so the fact that it's developmental changes related to the word versus non-word contrast makes a lot of sense. But of course, further studies will need to investigate this with study designs that are able to pull apart this even further. Okay. And lastly, for study 1, we did a quick check of the base rate to figure out the baseline response to the visual input. And we expect early visual brain areas to be involved. We don't expect condition effects. We did this by looking at the base frequency of all three conditions. We get this area of the brain here that is very similar to the early visual brain regions, and there's no condition effects. But we did not stop here. We found most researchers typically stop here. They find that, oh, there's no differences between the different conditions. And so they move on and they look at the more interesting aspects that I showed you in study 1. But we're not satisfied with that. And we wanted to dig deeper into this base level processing. We did, and we found something new and interesting that was not found in previous studies. We got a second reliable brain region that seemed to overlap with the VWFA, but it's more bilateral. And this brain region also has no condition effects. But this is an analysis of the base rate. So why is it that we're getting processing in a brain area that is doing processing later on than just early visual? Perhaps something else is happening more here. Perhaps the slowing down of the presentation rate is allowing for something more to happen. So I wanted to study this further, and this led us to study 2. And for study 2, we're looking specifically at the latency or the timing of the neural response. It's motivated by the idea that you can look at the same data set from different perspectives. When amplitude is telling you one story, the timing of the response may be telling you a different story. Together, the multidimensional approach helps us pull out different insights into individual variation found in children. In study 2, I will show you how I was able to calculate latency using a novel method. And I will show you for the first time ever how I was able to get latency values at the individual participant level using this method. And this was not something that was ever achieved in the past. So the methods of study 1 is very similar to study 2. Data collection is the same, which it just differed in the way we analyze the data. Skip through that.
Trang, I just wanted to note we have 10 minutes left.
Okay.
And I'll give you a short 5 minutes.
Okay, thank you. Okay, so that's good because then I'll spare you all the math that I was going to show of how latency is calculated, and I'm just gonna go straight to the latency values that we got out. So I didn't have the time to show you this, but it's really cool that we were able to get individual latency values, and that the latency values also correlated incredibly with each other. This means that the different parts of the brain are like dancing together at a very precisely same rate, and they're all reacting or responding to the visual stimuli that's presented at a delay of about 160 milliseconds. And that these individual latencies that we were able to get correlated with age for all 3 conditions. What we're seeing here is that whenever I circle, it means it's significant. We see that older children have a lower frequency. That means that their response to the brain's response to the visual stimuli is faster. And this is consistent across the ages. And what about reading? You see the exact same pattern here. The x-axis is a reading score that we collected, and the y-axis is the latency. And across all 3 conditions, children with higher reading scores also have faster processing or like faster timing response in the signal. It's like as the children are getting older or they're becoming better readers, their brain waves are also dancing much quicker and responding much quicker to the beat. So in summary, here are the 7 key findings of my 2 studies. Finding 1 is that we found robust signal to letter form processing, and this is the first to have been detected in 3rd to 8th grade children. New finding 2 is that we found the word form processing signal, which is the first to be ever detected in children. We found that letter form and word form processing is not a simply additive process, but that maybe the word form processing in the word versus non-word contrast may be more salient than the word versus pseudo-font contrast. 4, we found that amplitude decreases significantly with age, and this occurs in 2 very distinctly different ways in 2 brain regions. 5, we found that the base rate activates brain region that suggests more than it's just doing early visual processing. We found individual latency estimates for the first time ever. And we found that latency is negatively correlated with both age and reading. And for future research, we can go on to investigate how these different factors that we found here changes over time using a longitudinal study. We can look at even younger children like K2. Actually, I was also involved in a study that looked at K2 and investigate children with reading and learning disorders. So yeah, that's the end of my presentation. I would like to thank everyone. In my presentation, I used we a lot instead of I, even though I know it's proper in a dissertation presentation to say that I did all of this, but it takes a village to raise a PhD student, and this is my village. So thank you everybody in the Education Neuroscience Lab. Thank you to my family and to my friends for all the support that they have given me over the years. And most importantly, I would like to thank my main advisor, Dr. Bruce McCandless, for giving me the freedom to explore my academic interests, especially during the first few years of my PhD program, and for believing in me in moments that I doubted myself. So thank you.
I'd like to invite everyone to unmute and give Trang a great round of applause.
Woo!
And open up about 5 minutes for questions from the audience, not from the committee members.
I think I can go. Frank, thanks for great presentation. And I'm now going to ask you more about application. So what you see, like all these amazing findings that you have, can you just share with us like some suggestions or recommendations that we can use now, for example, for the classroom that should be applied there?
Oh, that's a good question. I think this research is very on in the very early processing. And so I think a lot more work will need to be done before application in the classroom. I think the design and of these projects were made for the future possibility that it could be used for the detection of early reading difficulties. So if you're able to have a tool that can detect letter form and word form processing in children without them having to do a particular task or in a short amount of time, then that was ideal for early assessment and diagnosis of disorders like dyslexia. So that's the direction at which we went about designing the study. And I would love to see future studies that are able to look at that, to develop that further. Any other questions?
Trang, that was fantastic work. Congratulations. I tried to find the raise hand feature, but I couldn't. You talked about a bunch of different potential directions to take the work, and it's really exciting. If you were to continue with this, what would you want to do first? Like, what would be the first set of questions you would ask next?
I would want to be able to study children with dyslexia, and actually that was what I had initially planned. We had a partnership with a dyslexia school, and we had a lab all set up and ready. We had already collected behavioral data on the kids, and then we were scheduled to collect EEG data on them starting from Monday, and we would be done in like 1 or 2 weeks. And that Monday also happened to be the day that COVID happened and everything was closed down. So that didn't work out, but that would be the first direction that I would go with.
Awesome, thank you. Yeah, this pandemic has really thrown a wrench in things, huh? But really, once again, fantastic work.
Thank you, Ben, and thank you for helping me with parts of the study.
I think we have time for one more question before we move into closed session.
He'll bite. Please ask questions.
Okay, great. Thank you all very much for attending this public session of the oral defense for Trang. And at this point, we are going to invite the committee members to remain in the Zoom room. Those who are nearby and want to join in person can come join us in Sarah's Hall. And otherwise, we will continue the closed session. And thank you all for coming. And congratulations, Trang, on a great presentation.